Welcome to your first semester of cell biology. Today, we are going to explore one of the most critical structures in any eukaryotic cell, the nuclear envelope. Think of this not just as a simple bag holding your DNA, but as a highly sophisticated, heavily guarded fortress. Understanding how this fortress is built and how it controls what goes in and out is the absolute foundation for understanding how complex life survives and functions. Let us dive in. To truly understand the nuclear envelope, imagine a bustling medieval city filled with workers, factories, and constant chaotic activity. In the very center of this city sits the king's chamber, holding the master blueprints for everything the city needs to survive. To protect these irreplaceable blueprints from being damaged by the chaos outside, the city builders construct a highly secure, double-layered stone wall around the chamber. This protective wall is exactly what the nuclear envelope is for a living cell. It ensures your precious genetic information remains safe, organized, and perfectly controlled. Now that we have our city analogy, let us look at the actual biological structure. The nuclear envelope is a complex double membrane structure that completely encloses the nucleus of eukaryotic cells. Its primary job is to physically separate the inner nucleoplasm from the outer cytoplasm. It consists of two distinct lipid bilayers. The outer nuclear membrane is actually continuous with the rough endoplasmic reticulum, and just like the rough ER, it is studded with protein-making ribosomes. This shows how deeply interconnected the cell's organelles really are. Moving inward from the outer membrane, we find the inner nuclear membrane. This inner layer binds to a supportive protein network called the nuclear lamina, which acts like a structural scaffolding to keep the nucleus from collapsing. Between these two membranes lies the perinuclear space. This is a narrow, fluid-filled gap that connects directly to the internal lumen of the endoplasmic reticulum. So the envelope is not just a wall, it is a dynamic, fluid-filled double barrier that provides immense structural integrity to the cell's command center. We know the wall is secure, but how does the cell communicate with the inside? The envelope is heavily perforated by massive, intricate protein structures known as nuclear pore complexes, which span across both the inner and outer membranes. These act as selective security channels. They strictly regulate the bidirectional transport of large macromolecules, such as RNA and proteins, deciding exactly what can pass. Meanwhile, they allow smaller molecules and ions to diffuse freely. Together, these pores create a highly dynamic and selective barrier, essential for genome integrity. Let us visualize everything we just discussed. On screen, you are looking at a cross-section of the nuclear envelope. Notice the top region, the cytoplasm, and the bottom region, the nucleoplasm. Separating them is the double lipid bilayer. You can clearly see the outer nuclear membrane, studded with ribosomes, and the inner nuclear membrane resting on the mesh-like nuclear lamina below it. Between the membranes is the perinuclear space. Right in the center, spanning the entire structure, is the nuclear pore complex, acting as the guarded gate for molecules traveling in and out. Having seen the structure, we must ask, why does this specific barrier matter so much to our survival? Without the nuclear envelope, eukaryotic life as we know it would be completely impossible. If there were no barrier, the processes of reading DNA and building proteins would happen simultaneously in the same chaotic space. By physically separating these two processes, the nuclear envelope allows the cell to carefully process, edit, and proofread RNA messages before they are ever turned into proteins. This separation is a crucial evolutionary step in preventing catastrophic genetic errors and diseases like cancer. This evolutionary protection leads us directly to real-world medical applications. Understanding the nuclear envelope is absolutely vital in modern medicine. For instance, mutations in the structural proteins of the envelope, like the nuclear lamina, cause severe premature aging diseases, such as Hutchinson-Gilford progeria syndrome. Furthermore, dangerous viruses like HIV must figure out how to bypass this exact envelope to insert their viral DNA into our genome. By studying how this barrier functions and how it can be breached, scientists are able to design much better, more effective antiviral drugs. While the nuclear envelope is a biological structure, we can use simple mathematics to understand how efficiently it works as a transport barrier. This brings us to our primary transport formula. Q equals N times R times T. Conceptually, this tells us that total traffic depends on how many gates are open, how fast they work, and how long we watch. Q is the total quantity of molecules successfully transported. N is the total number of nuclear pore complexes acting as open gates. The variable R is the specific transport rate of a single pore, measured in molecules per second, and T is the total time elapsed in seconds. If a cell needs to export more RNA, it must increase either the number of pores or the transport rate. Now that we have our formula, let us apply it to a real biological scenario. Imagine a typical mammalian cell that is actively dividing. We are given that the nucleus has exactly 3,000 nuclear pores. We observe that each individual pore can transport 10 mRNA molecules per second. We are asked to find the total mRNA exported in a time span of 60 seconds. We choose our transport formula because we have N, R, and T, and we need to find Q. We substitute 3,000 for N, 10 for R, and 60 for T. Multiplying these together, we arrive at 1,800,000 mRNA molecules. This staggering number highlights the incredible efficiency and high-traffic nature of the nuclear pore complexes in just 1 minute. That massive transport volume we just calculated leads us to an important physical constraint. A cell cannot just have infinite pores. Transport capacity is strictly limited by how much surface area is available on the nucleus. For a perfectly spherical nucleus, we calculate the total surface area, A, using the geometric formula 4 times pi times r squared, where r is the radius. Once we know the surface area, we can find the total number of pores, N, by multiplying the area A by the pore density, represented by the Greek letter rho. Rho tells us how many pores are packed into each square micrometer. If the radius increases, the surface area grows exponentially, allowing the cell to support many more pores. Before we can find required transport rates for a specific cell, let us see this geometry in action with a concrete example. A researcher is studying a yeast nucleus with a measured radius of 1.5 micrometers and a pore density of 12 pores per square micrometer. We need to find the total number of pores. First, we plug the radius into our surface area formula. Squaring 1.5 gives 2.25, which we multiply by 4 and pi to get approximately 28.27 square micrometers. Now that we have the total area, we multiply it by the density of 12. This gives us approximately 339 total nuclear pores. We round to a whole number because a cell cannot have a fraction of a pore. So we have proven that this specific yeast nucleus has exactly 339 pores available. This brings us to a critical physiological question. Can it survive under stress? Let us set up the derivation. The cell needs to export 50,000 ribosomal subunits in a strict time limit of just 10 seconds. We know our total pores, N, is 339. We start with our standard transport equation. Q equals N times R times T. Our goal in this chunk of the derivation is to isolate the unknown variable, which is the transport rate R, to see exactly what burden is placed on each individual pore. Continuing our derivation, we algebraically rearrange the transport equation to solve for the rate R. We do this by dividing the total required molecules, Q, by the product of the number of pores, N, and the time, T. This rearranged formula perfectly illustrates how the burden of transport is shared equally among all available pores. Substituting our known values, we divide 50,000 by the product of 339 and 10. This yields a final required rate of approximately 14.75 molecules per second. This beautifully demonstrates how mathematical modeling predicts survival. Each pore must process about 15 subunits every second, or the cell will fail to assemble ribosomes and die. We have covered a tremendous amount of ground today. Let us review the key takeaways. First, remember that the nuclear envelope is a highly sophisticated, double-membraned regulatory fortress, not just a simple bag. Second, it relies on the nuclear lamina for structural support and nuclear pore complexes for highly selective transport. Third, this physical separation is the defining feature of eukaryotic life, protecting against catastrophic genetic damage. Finally, we saw that transport capacity is mathematically governed by pore count, rate, and time, proving that the physical size of an organelle directly dictates its functional capacity. You now have a profound understanding of how our cells adapt and survive every single day. Excellent work.